Okay, let's go ahead and get started. Welcome to Medicine Grand Rounds. I am going to first introduce Eric Opersla to you, who is the Division Head of Infectious Diseases, who will introduce the Tim Gill Lectureship.
Thank you, David. So a quarter century ago, in the midst of another great pandemic emergency, HIV-1, Tim Gill, who's a Colorado entrepreneur and philanthropist, endowed the Tim Gill Professorship, which I'm honored to hold. And as a corollary to his gift, Tim also endowed an annual visiting professorship. And this allows us to invite prominent scientists to speak about their HIV research each year. And this amazing pandemic year, we're delighted to have Dr. Jeremy Luban from the University of Massachusetts as the 2020 Tim Gill Visiting Professor. David?
So Dr. Luban is a professor in the program in molecular medicine and in biochemistry and molecular pharmacology at the University of Massachusetts. He's the David Freelander Chair in AIDS Research. He received his undergraduate degree from the State University of New York in Purchase and his MD degree from Columbia University. Went on to residency and fellowship at Columbia Presbyterian, and then stayed on as faculty at Columbia. Spent 4 years in Switzerland at the Institute for Research Biomedicine and the University of Geneva, and then came back and has been a faculty member at the University of Massachusetts since his return from Europe. Jeremy has done absolutely spectacular work in HIV, and I'm really pleased that he was chosen as a Tim Gill lecturer. He is a central figure in understanding the HIV life cycle, and he's focused on how HIV acts when it gets into the cell, and what are the factors that allow HIV to enter the nucleus of the cell. And this has led to spectacular discoveries that are sure to change the way we treat individuals with HIV. He's been recognized for these discoveries in terms of being elected to the American Society for Clinical Investigation, the Association of American Physicians, and he just recently received a merit award from the National Institute of Allergy and Infectious Diseases. What I'd like all of you to do is put your questions in a Q&A box, and I'll moderate the questions at the end of Dr. Luban's presentation. Dr. Luban.
Thank you, David. Thank you, Eric. Thanks for this incredible opportunity. And I just wanted to point out, I just saw about an hour ago, interesting news given the Gill Foundation's efforts. I think this is an event of really historic proportion. This call from the Pope to recognize same-sex couples. So anyway, it's really an honor to be able to speak with you today. I'm going to try to convey to you some of our excitement about HIV and a little bit of what we're doing with SARS coronavirus 2 and within a specific clinical context, and that is to look at the effects of these viruses or the interactions of these viruses with innate lymphoid cells. And so these two viruses, HIV and SARS-CoV-2, are zoonotic. They've presumably, we believe, both come to people from animal reservoirs. Over the course of the talk, I'm going to give you a very brief history of HIV from my particular molecular biology perspective. I'm going to talk a little bit about problems of chronic inflammation that have been reported with HIV infection despite highly effective antiviral therapies. I'll take a minute to tell you the little bit that I know about what innate lymphoid cells are and why they might be relevant to discussions of inflammation and viral infection. And I'll show you some evidence that ILC abnormalities at least accompany HIV infection and may explain some of the inflammation that is seen with HIV infection. Then I'll tell you a little bit about SARS-2. I'll tell you about some work we're doing to try to understand the effects of age, sex, and the virus itself on innate lymphoid cells in the context of COVID-19. So first of all, I want to remind you, this is the going to the HIV history to tell you that the appearance of AIDS in the beginning of the 1980s was a really terrifying experience to live through. I'm a New Yorker. I was in New York, and it was quite evident that something very dramatic was occurring. I was just finishing college. I was working in a lab as a lab technician, and there were these reports of this disease that was killing people who were otherwise perfectly healthy, or at least they had no explanation for why an opportunistic infection like pneumocystis would be killing them. There was, I would say, at least in New York at that time, pneumocystis was probably the most common serious hospital presentation that we would see. There were times when I was on call at Columbia where we would admit 4 or 5 people with chest x-rays like this in a day. It's in many ways very reminiscent of what's happening today with COVID-19, except that the demographics were quite a bit different. So the history of the outbreak is really interesting to review. It's a history that involves epidemiology, it involves technical innovation, all kinds of things play into the outcomes that we witnessed. So the first report was in 1981. Already by 1984, the causative agent was discovered. There was a lot of resistance from multiple camps about whether this actually was the cause of the disease, but I think there's really no doubt evidence from multiple sources, different kinds of experiments, epidemiologic data. There's no doubt it's the cause of this disease, AIDS. By 1987, the first drug was approved. I remember giving this as a single agent in the clinic to patients I was following who were HIV positive. And by the mid-1990s, the first effective antiviral cocktails, usually with 3 combinations of 3 drugs, viral protease inhibitors and RT inhibitor combination. This was a real game changer and completely changed the demographics of the disease, shifting it from being a certain death sentence, shifting it from being an in-hospital problem to an outpatient kind of chronic disease. So today, there are very potent drugs that suppress viremia. There are new drugs being developed all the time, including on your campus. There's a beautiful report about a new long-acting capsid binding inhibitor from Eric's ID division. It's possible to protect the CD4s from being depleted, and it's possible to prevent AIDS. Now, I use the word miracle in ironic sense, but I just want to emphasize the fact that miracles don't just happen. the miracle of the response to AIDS and the discoveries of its cause and the development of the therapies were able to happen because of a very, very long history of fundamental biological research. So retroviruses are among the first viruses that were described in the biological literature. People studied them at the very beginning, the first decade of the last century. And this is a photograph of a group of a lot of prominent retrovirus investigators that took place roughly corresponding to the 100th anniversary of the first descriptions of retroviruses. This was in Prague. Jan Svoboda, who's shown in the working in the hood at the age of 80, organized this meeting. So last year, I think it was last year, time is very confusing with the whole COVID situation, but March 2019, there was a statement about the US plan for dealing with the epidemic going forward. And this was signed primarily by Tony Fauci and colleagues at the CDC as well. And the 4 pillars were that we had to diagnose people as early as possible. That actually is an issue that was debated for many years, but ultimately was shown that it was important to get people diagnosed and treated quickly. Pillar 2, treatment. And there are many reasons why you want to treat, not only to maintain the health of that individual, but to prevent transmission of the virus. You want to prevent at-risk individuals from getting infected, and there are ways to do that now with pre-exposure prophylaxis. And then finally, of course, we want to rapidly detect and respond to any clusters or changes in the epidemiology that look threatening. But these all, to a clinician who takes care of people with HIV, these all seem really obvious. And I think there are some things that we have to consider that are not addressed here. So first of all, the epidemic from day one has always been very slanted in terms of the groups that are being hit hardest by this infection. And this is the latest pie chart from the CDC showing that indeed still today, years after the start of the outbreak, we still have a really high, a disproportionate burden on certain parts of the population. This clearly is a disease of poverty, and it tracks with that around the world, and it's also very specifically affecting a really large proportion of the African American population. So I think what this statement from Tony Fauci doesn't address is an important point, which I think is something that really has to be faced in our country, which is that healthcare should be a human right and shouldn't be thought of as a commodity, which is the way that our whole medical system and all of our legal system views medical care. I just want to remind you that it was actually FDR way back when he was president who came up with the idea that healthcare was actually an essential human right. He wanted to incorporate that into a kind of second Bill of Rights, which never happened because he died prior to that. But his idea was then brought to the United Nations as it was formed. It was argued over extensively, but Article 25, which the US signed at that time, does indeed espouse the essential right to health of all people on planet Earth. And years after that, Martin Luther King, in a pretty major speech of his many, pointed out this really telling statement that of all the forms of inequality, injustice in healthcare is perhaps the most shocking and inhumane. And I was looking for points about the fact that healthcare is a human right. And when I searched for it, I came across this very interesting article of all places. The American Bar Association website had this statement, very, very critical of the US healthcare system, which states explicitly, by codifying a system allowing huge corporate profits on healthcare as a commodity, our government has actually impaired, not improved, our right to healthcare. And I think the big piece that's missing in the whole conversation that was not addressed in that article from Tony is the economic and class disparity in our country that's not part of the conversation, whether it's the DNC or the GOP that we're talking about. This is data showing that the economic growth by income percentile in the United States, well, up until the time that Obama was still president, this is before Trump was in the White House, and you can see that there's a really extraordinary growth in the economy of those at the top of our society. So people living with HIV are a growing population. There's maybe 38 million worldwide and over 1 million in the United States. There's still quite a few new infections per year. There have been major advances in terms of getting therapy to people around the world, largely thanks to George Bush's funding and efforts. In the US, the latest estimates are that there's something like 38,000 new cases in the USA. And when you look at the demographics, it's kind of interesting how it's shifted very much to the southern parts of the country from the Northeast and the West Coast. This is kind of an interesting paper that I think is pretty important to think about. This is a study that was done in the Netherlands. And this group estimated that by the year 2030, which is only 10 years away, an enormous proportion of those who are HIV infected are going to be over 50 years old. I think it's really interesting to think about that. And another point that the literature has documented, there are many, many studies about this, is that HIV infection seems to be associated with a relative risk of cardiovascular disease that is on a par with the standard well-known risk factors for cardiovascular disease like smoking and hypercholesterolemia. So I think this is something that's, this is a topic, the aging HIV population, the inflammation that is associated with HIV infection and the relative risk of cardiovascular disease associated with that has to be considered. So why is there this increased risk of cardiovascular disease in people living with HIV? I think there are a number of possible explanations that have been proposed, and there's good data supporting. Some of the medications actually change lipid metabolism, some of the antiretroviral therapies. Dolutegravir, one of the, say, newer anti-HIV medicines that has become widely used, an integrase inhibitor, was shown last year to be associated with much higher rates of obesity. And I think these are both studies that were done in Africa. So there's been a history of different drugs having different metabolic effects, and these are all potential, each of these are potential explanations for this risk. Another theory that's been in the literature for a while now is that when you look for markers of gut epithelial integrity, whether you look at the gut itself or you look in the plasma for markers of molecules that may have leaked from the gut into the systemic circulation, there is evidence that in many people with HIV, there is loss of gut epithelial integrity. Perhaps you actually have microbial leakage or leakage of microbial molecules that stimulate innate immune signaling pathways systemically and contribute to systemic inflammation. So how does HIV disrupt gut epithelial integrity? We know for sure that HIV, when it first enters the body, it destroys the CD4 positive T cells, the CCR5 positive, CD4 positive T cells that are in the lamina propria and the tissues associated with the gut. But we've wondered whether particular cells called innate lymphoid cells that are present in the lamina propria and have been shown in mouse models to be very critical for maintaining the integrity of the epithelium, whether these cells might be perturbed by HIV infection. And I'm very excited to be meeting Steph Dillon later this afternoon, who hopefully will teach me about this topic, something I'm just beginning to learn about now. So what are innate lymphoid cells? So I think most of you are probably familiar with T cells and the fact that they come in different flavors, that they can be divided up into CD8 cells with killer activity, and then CD4 cells with different flavors of helper activity. And these cells are, each of these are defined by particular transcription factors that promote their development. And then they're characterized by particular cytokines that they produce. And so, for example, Th1 cells make interferon gamma that promotes antiviral responses, whereas Th2 produces cytokines such as IL-4, 5, and 13 that promote responses to parasitic infections, for example. And then each of them have their complications, their inflammatory consequences, allergic consequences that they promote. So the same way we have these different lineages and flavors of T cells, there are very analogous subsets of innate lymphoid cells. So something called the natural killer cell or NK cell is a functional homologue of CD8 cells. It kills by similar mechanisms to CD8s. And then there are these sort of T helper type innate lymphoid cells that have the same kind of transcriptional program and the same kind of cytokines that they make. These cells differ from T cells in that they do not have clonotypic antigen specific receptors like the T cells do. They make cytokines, they respond to the environment, they promote the well-being of the various tissues that they reside in. They're often at barrier interfaces in the mucosa, in the gut, in lymph nodes, in the lung. And there's very interesting data from mice, very little so far from humans, showing that they play very important roles in the response that people make when challenged with, for example, influenza virus. So if you disrupt the ILC2s that are present in the lung of the mouse and you challenge with influenza, the disease is much more severe. So we wondered whether HIV might be playing with these. The ILC3s are the flavor that is present in the lamina propria of the gut. And we focused on those, as I'll show you. The blood ILCs tend to be ILC2s in their nature, or the actual NK cells. So identifying these cells is not trivial. You have to, they're usually identified by flow cytometry. You have to gate out all of the more common, better known cell types like T cells and B cells and monocytes with a whole panel of markers. You gate those out, and you're left with the so-called lineage negative cells. NK cells have specific markers that you can use to distinguish them from the cytokine producing innate lymphoid cells. And then there are particular markers that are positive indicators of the presence of innate lymphoid cells like the IL-7 receptor alpha or c-KIT CD117, and you'll see some data with some of these. They can then be broken down into subsets based on the transcription factors or other markers that they bear. So this is one set of data looking at the UCSF SCOPE cohort. This is working with Steve Deeks. And we looked at the ILCs that we can detect in the blood. These are ILC2s, in fact, in terms of their flavor. And what we are comparing here, the percentage among these lineage negative cells in HIV negative people, in people who are viremic, who are not being treated with antiretroviral therapy, in people who are on therapy, and in people who are spontaneous controllers. These are not elite controllers, but these are spontaneous controllers. And in each case, each of these groups, the innate lymphoid cells are indeed reduced. And when we've looked in more detail at blood samples from here in Worcester, and we see that the ILCs tend to be lower, whatever the viremia level is, or whatever the CD4 counts are. So it seems to be an independent effect of HIV. If you look in the plasma for markers of inflammation, like soluble CD14, this is something that monocytes make. It's associated with cardiovascular disease, coronary vascular disease in people who are living with HIV, and predicts subclinical atherosclerosis in studies with people who are HIV positive. We indeed see an inverse correlation. That is, if your ILCs are lower, you have higher levels of this inflammatory marker, and this has been seen with other markers of inflammation in the plasma as well, suggesting that the abnormalities that we see with ILCs may have a functional consequence. So then we started looking in the gut of people who are HIV positive. We were fortunate to work with a gastroenterologist here and collected biopsies from people who are undergoing screening colonoscopy as a result of their age. It's recommended if you're over 50 that you have this done. It turned out these cells were very delicate and had to be processed immediately. We were unable to freeze them, so we were lucky that we had this connection and we could go directly from biopsy to the lab. We found that the results were similar in sampling all across the colon up to the terminal ileum. And this just shows our gating, looking at the CD127 positive and c-KIT positive cells out of the lineage negative subset. These cells in the gut are ROR gamma T positive, which defines them essentially as ILC3s, and they're capable of making IL-22. And when we look at the effect of HIV in this group of, I think it was about 7 HIV positive people compared to HIV negative, they indeed had reduced numbers of these viable lineage negative ILC3 type cells in the colon lamina propria. And all of these people had undetectable viremia when these biopsies were taken. Working again with these were samples from UCSF and looking by immunohistochemistry at the presence of c-KIT positive ILCs in the lamina propria of the gut. What we see if we compare HIV negative to positive samples is that there is a depletion of these green dots of these c-KIT positive cells in the gut that corresponds to a loss of ILC3s. There was an interesting observation, at least for me, I was not aware of this, that we would often see in HIV positive samples, these clusters, these lymphoid aggregates. This is staining with CD3. These are T cells, presumably, that we did not see in the HIV negative samples. And I think this is something that would be very interesting to look at in more detail. Quantitation of this immunohistochemistry shows that indeed the signals from these ILCs are decreased in these HIV positive people who are on therapy. So even if the viremia is inhibited, the ILCs are depleted when we look at the fresh isolated biopsies or if we look by immunohistochemistry. And accompanying this, we could see markers of inflammation in the tissue. This is one of these lymphoid aggregates, again, showing a very strong signal for a protein that is induced by type 1 interferon, which we never saw signals like this in the controls. And as well, if we looked for neutrophils, we would see neutrophils in the HIV positive samples that we just don't see, or very, very rarely in the HIV negative samples. And this is quantitation of that, showing increased inflammation marker and neutrophil marker in the gut. So how would HIV affect these ILCs? We don't think that it infects them. And I say that because from extensive sequencing that we've done of these cells, we don't see the primary receptor CD4 or CCR5 expression on these cells. We don't see these proteins if we look by flow cytometry on these cells. We've pulled these cells out and attempted to infect them with full virus, with vectors, and we've been unable to detect infection with the standard assays that we use in my lab. So this would suggest that whatever's depleting these cells is acting indirectly and is not directly killing them, perhaps the way that the virus does when it eliminates CD4 positive T cells. So we incubated ILCs. These are blood ILCs in the dish with a whole bunch of stimuli, whatever we could get our hands on. These are just some of the many things that we looked at. And the only thing that we found really annoyed these cells were cytokines that act through the common gamma chain. So these are a class of cytokines that include IL-2, 4, 7, and 15, some of which are present at extremely high levels in the plasma of people during the acute stages of HIV infection, and could possibly explain how these cells are eliminated. It turns out we can prevent the loss of these cells by stimulation with IL-15 by blocking with a JAK3 inhibitor, not other inhibitors. And JAK inhibitors are seeing increasing usage in all kinds of inflammatory disease. You could imagine that maybe in some context, it would be possible to preserve them by targeting this pathway. So in terms of the ILCs in people living with HIV, we see that they're depleted in all of the people living with HIV, whether they're on therapy or not, that we can see. We've not yet looked at elite controllers, and that's something that is in the works. We have some samples we're going to look at, again, from Steve Deeks at UCSF. I've shown you that the loss of these cells seems to correlate with increased infiltration of neutrophils and interferon stimulated gene expression in the cells that are present, and as well with systemic markers of inflammation. In vitro, we can mimic disruption of these cells by stimulating them with common gamma chain cytokines, and we can block that with JAK3 inhibitors. Our interpretation of these results is that the reduction in ILCs is permanent. There is one study out of Durban that suggests that if you catch people early enough in the acute infection stage, giving them antiretroviral therapy, I guess during the acute cytokine storm that occurs with the acute infection, you may be able to preserve the cells, at least as gauged by looking at the ILCs in the blood. And so we postulate that their loss, their disruption is due to some effect of these acute cytokines, and that disruption of these cells in the gut by rendering the epithelium more permeable. ILCs make a number of factors that stimulate epithelial cells to maintain their integrity of their junctions, and also to secrete antibacterial peptides as well, that perhaps because of the leakage that results, you get this chronic systemic inflammation. So there are a number of questions we're interested in looking at going further. We'd like to know in more detail what precursor stage is depleted. Is it just these sort of end stage innate lymphoid cells, or are earlier stages disrupted? We'd like to know why they don't come back. So we can see depletion of the ILC3s in the lamina propria, even when we see CD4 cells coming back in many of the samples. And are there ways that the loss could be prevented or even restored in such a way that you could prevent the complications of this chronic inflammation? So I'm going to jump real abruptly and tell you about SARS-CoV-2. I want to start by showing a picture of June Almeida here. She dropped out of school when she was 16. She's from Glasgow. I believe she went to London, started working there, learned some technical skills, and then had a position in Canada for a while, doing microscopy, developing techniques for imaging viruses by microscopy. And then she went back to London, where she was contacted by the people from the famous flu center in Salisbury, UK, where they were challenging, they would essentially collect snot from people and challenge volunteers in a controlled setting and see if they could transmit the common cold to people. This was an effort that came out of World War II with funding from the UK, funding also from the US largely. And the idea was that the common cold, as modest as it may be to the individual, it can have a big toll on an army if it's incapacitating its soldiers. And I think that was probably the initial impetus for setting up this program. They discovered a lot of viruses. They discovered how viruses are transmitted through the nose. And at one point they had a sample that they couldn't figure out what it was. They couldn't characterize it. They brought it to June, and she saw this very distinct shape that we now know with our current outbreak as a coronavirus. Interestingly, she tried to publish this for a couple of years, and the reviewers rejected her papers, saying that what she was showing them were poorly fixed influenza viruses. Nonetheless, we now know that these are coronaviruses. There are 4 that circulate around the world that we've all been exposed to, and since then, we've had a couple of really serious deadly ones: SARS coronavirus 1, MERS, and now SARS coronavirus 2. Just my own personal interest, June, at the end of her career, actually took pictures of HIV. These are really remarkable images of the cores of HIV-1 virions. So I don't know, you're probably all as familiar or more familiar than I am with the current outbreak. It's bad, it's a mess, a lot of cases, a lot of death. Again, there's enormous disparity in terms of who's getting sick, who's going to the hospital, and who's dying. Very similar pattern. This is data, the rates per 100,000 people by race or ethnicity. You can see again, the rates are much higher for African Americans than they are for white Americans. And this correlates with all sorts of economic indicators. So again, one has to ask, does it make sense to link health insurance to employment? What is the connection there? Why isn't there universal health coverage? And one has to consider the disparities in economics that are causing this kind of health disparity. So in Massachusetts, there's been an incredible effort to respond to the outbreak. It's called the Massachusetts Consortium on Pathogen Readiness. And through this consortium, we were able to obtain blood samples from people who were admitted or followed at Mass General Hospital. And this is the cohort. I'd say there are several previous reports, some published, some in bioRxiv, indicating that there are abnormalities in different subsets of cells in the peripheral blood mononuclear population with COVID. And our cohort, when we started to look at it in detail, really bugged us because it's extremely slanted, not well balanced in terms of age and other features that are known to have big effects on the outcome of SARS-CoV-2 infection. And I would say that I'm not really aware of any paper so far that has, what I would say, adequately addressed the confounders of age and sex. And so we attempted to model the effects of age and sex in the context of COVID infection to try to see what was going on. And so what I mean by unbalanced cohorts, so first of all, we have a SARS-CoV-2 negative population that we've collected that spans the ages. There's a hospitalized population, which is generally on the older side, as everyone has reported. And there were SARS-CoV-2 positive people who were treated as outpatients, which are clearly slanted on the younger side as compared to this. There's also a big shift in the gender. So the hospitalized population, 60% were male, whereas the outpatients, only 24% were male. So these are very preliminary analyses that we're doing, and this is an analysis just of the SARS-CoV-2 uninfected controls. So this is not COVID-2 infection. Looking at the effects of age and gender, so female are red, male are blue. And there are a couple of interesting things one can see. This is our regression analysis of this data. First of all, CD4s, CD8s, and innate lymphoid cells decrease with age, independent of COVID. And that you can see with this slanting, this down slant in the ILCs, the CD4s, and the CD8s as people age. And this data is all shown in log2 so that it's much easier to compare the different parameters and also to model the effects. And one of the really interesting things about the effects of age is that while CD4, ILC, and CD8 are decreasing, you'll notice the negative sign here, and this is the log2 exponent decrease of CD4 per year of life in an uninfected person. In contrast, the NK cells, or specifically shown here, the CD16, these are FC receptor positive NK cells, these go up with age. And that is something that we thought was pretty striking. The other thing is that, well, CD4s and ILCs are lower in males, and you can see that the difference between the red line and the blue lines here. This is not the case, at least not statistically significantly so, with CD8s or with the NK cells. So we wanted to then look at the effect of COVID on this kind of background of effects that are occurring just because of the demographics of the starting population. And so what this shows you are the actual numbers of CD4 positive T cells or ILCs per million live cells in uninfected, in hospitalized, or the outpatient treated SARS-CoV-2 people. And what you can see is that in the regression models, if you account for age, which has effects on all of these populations, and if you account for whether a person is male or female, which has clear effects on the CD4 and the ILC populations, the hospitalized patients have really big effects just on the ILC and on the NK cells. So independent of the effects of age and sex, the ILC cells and the NK cells are altered by COVID-19 infection. And so, again, this is very preliminary work. We're just analyzing this now. One has to wonder, what is the significance of these reductions that are seen with age and with sex in these populations of cells? And what is coming first? Are these things setting people up so that they're more likely to have the complications, or to what extent are these reductions a consequence of having the infection? And it seems that the main effect of the infection itself is on innate lymphoid cells rather than on the T cells. So I'm going to wrap up now. I'm just going to tell you very quickly about a story that isn't pressed now, about this amazing virus. It's amazing from the standpoint of molecular biology. One of the incredible things about this outbreak is that there are people sequencing the viruses all over the planet. Latest tally at this website is sequences from 155,000 isolates from all over the world. This has allowed the outbreak to be tracked. It's allowed us to look for interesting variants. And one of those variants is this D614G variant, which is shown in yellow here. It wasn't present in the virus that started the outbreak. We don't know if it first appeared in Shanghai or in Bavaria, but those are the places where it was first sequenced. And it's now gone to fixation. It's essentially the only form of the virus that you see around the planet. We wondered what it was doing. We tested it in our lab, and comparing the variant to the ancestral form, we found that on various infection models, the variant is clearly more infectious. We've characterized this in detail, and ultimately, we think the most interesting thing about the mutant is its structure, which Quang Shen, my colleague here, solved by cryo-EM. It has a much more open conformation. The spike protein exists in a closed or an open conformation, and only the open conformation can bind to ACE2. The D614G is much more in these fully open conformations, which we believe are primed to bind to ACE2 and fuse, makes it more infectious. There is data now in animal models that it's more transmissible. The good news, though, is that perhaps because it's more open, it's not resistant to therapies that are being developed. Specifically, we tested it against the monoclonals from Regeneron, and they're equally potent on the variant as on the original. But I think this points out that we really need to keep monitoring the sequences and look for surprises along the way, which perhaps would be selected for by some of the treatments that are now exerting selective pressure on the virus. And so this is my group. The ILC work, the hands-on work is all done by Yatao, who's really kind of a miracle worker with primary blood cells. It's done an enormous amount of work with the HIV and the SARS samples. And Lanya did the infectivity experiments and the characterization of the D614G variant. And Noah has done the analysis of and the modeling of the SARS coronavirus work that I showed you. Quang Shen did the structure of the molecule. showed you at the end there, and all of our HIV work has been really dependent on collaboration with Steve Deeks. Jacob Estes and Jason Brenchley helped us with the analysis of our samples. And we've had funding for our SARS work from SCPR and our HIV work from NIAID. Thank you.
Jeremy, that was fantastic. You met all expectations, and there are a lot of questions that we have. So let me just start at the top. Not that these are the most important questions, but the ILCs in the gut. Why don't they repopulate after the acute infection?
I have no idea. It's really mysterious. They seem, in most respects, developmentally, they seem similar to T cells, and why they don't come back, I don't know. It may be that there's some more primitive, they reflect a more primitive state or an earlier ontogenic kind of step in development, and that they have to be set down early, and that's when things happen. And if you screw that up later, you can't fix it. There are ILCs that are required, for example, for the formation of lymphoid anatomy, and those are events that are established during development, and probably at least it's been very hard to try to recreate experimentally. So that's my head waving answer. I don't know. If I think it could be, there are many diseases where they're disrupted, many inflammatory diseases. I think there's going to be increasing need to understand these cells and to address them in human disease pathology, autoimmune diseases, maybe in cancer in certain cases. There are tumors that make factors that change them, small cell lung cancer, for example. There are all kinds of connections, and they're difficult to understand, they're difficult to study, but I think the tools now exist with the kinds of deep sequencing methods that we have, that we can characterize them better and find out where we need them and maybe how we could intervene.
So how representative are the ILCs in the blood of the ILCs in the gut?
So the cells in the blood are a different subset than the ones in the gut. It's interesting that they are both affected by HIV similarly. So the ones in the gut are ILC3s, they make IL-22. The ones in the blood are ILC2s that make IL-13. For example, the ones in the blood look more like the ones in the lung. And if those are disrupted in animal models, those lung ILCs, you have a much more severe infection in the lung with influenza, for example. We are attempting to trying to get cadaveric samples from people with COVID to see if we can detect changes in those, say, the lung tissues or lymphoid tissue or the gut.
So then tell us how you interpret that association between CD14 and ILC2 in the blood, if there's that distinction between the blood and the gut ILCs.
Right. So the soluble CD14 that I showed was in the context of HIV infection, and a number of groups have reported that. We were just following that lead from the literature. The correlation we saw was either with the blood ILCs or with the gut ILCs. So they all seem to track together. Exactly what the connection is, we don't know. The model we have is that when you lose the ILCs in the gut, you have leakage of bacterial products into the body systemically, and that those molecules like lipopolysaccharide can activate myeloid cells like monocytes and increased levels of something like soluble CD14. You may get increased C-reactive protein, other things that are commonly monitored in the clinic.
Mm-hmm. And another question related to the ILCs, but this relates to SARS-CoV-2 infection. Is there a depletion of ILCs in the blood because they're going to the organ, the lung, that's primarily infected?
Yeah, so again, I don't know what the relationship is between the blood and the tissues. We're making a huge assumption that they reflect each other. The only thing I can say is that what we see in the blood reflects what's going on in the gut in HIV. We would like to be able to make the same claims with the lung. But so far we haven't had access to tissue that would allow us to do that. What the connection is, are these cells that are leaking out from the organs, like lung ILCs that are leaking out into the blood? Are they cells that are being newly generated from bone marrow precursors? We don't know. I mean, these are all really important questions, each of which could be addressed. They're technically difficult, and I feel like we really need to do these in people, in humans, as opposed to mice, to get a better sense, because there are lots of differences between us. Like we don't have tail ILCs, for example.
Tell us more about the mutation rate in SARS-CoV-2 and how that might complicate vaccine development.
Right. So SARS-CoV-2 is an amazing molecular machine. It's the biggest by far of the RNA viruses, biggest in terms of the length of the linear sequence of its genome. So it's roughly 30,000 nucleotides. HIV is 10,000 nucleotides for comparison. Ebola is 19,000 nucleotides. There is a coronavirus that's 40,000. These genomes are so big that they are beyond the theoretical limit of what would be tolerated given the rate of mutation of the replication machinery. And that raised kind of red flags when people are looking at coronaviruses. And the really interesting thing is that coronaviruses, in contrast to all the other RNA viruses, have a specific machinery in its replication, in its replicase, that allows it to correct errors. And so much has been made about the fact that it has this repair machinery that corrects errors, and that its error rate is lower than other RNA viruses. So that's true. But so the rate of mutation is much lower than in flu or in HIV or Ebola, for example. But there is a mutation rate. There are mutations that accumulate at a modest rate. And there are enormous numbers of transmission events that are occurring. There are enormous numbers of people who are becoming sick. There are enormous numbers of people now who are receiving treatments, and each of these selective pressures might force the virus to change. All you need is one nucleotide change, and it can alter how pathogenic the virus is, how transmissible the virus is. So this D614G mutation has been shown now in isogenic comparisons. Single nucleotide change makes this thing more easily transmissible from one animal to another animal. So I think the technology exists and needs to be used to monitor this outbreak, because there could be surprises coming down the road. Most, if you were to bet, you would think, well, it's not in the interest of the virus to kill people so much as maintain itself. And maybe over time, it'll become less pathogenic. but that's a guess, and that's a gamble that some people are arguing that, but I would argue we need to actually know what the sequences are, and we need to have rapid assays that will allow us to assay the function of those variants in the lab, so we know whether they have potential to cause problems.
Mm-hmm. So related to that, the spike protein, you've shown like a dramatic effect in terms of the 614G mutation in terms of structure. What do you say to those that just argue that this is a founder effect?
So I've been working on this basic problem that you're asking me about with 2 Ebola outbreaks, the big West African one. The same exact thing happened there. There was a single mutation that dominated, that appeared early and then took over completely in the West African outbreak in the glycoprotein, the spike equivalent. And the same thing happened in the more recent big Congo, Democratic Republic of the Congo Ebola outbreak. Again, a glycoprotein mutant, single mutant. In the SARS-CoV-2, there's the single mutant. In any of these cases, these could just be an event that's happened by chance, and they have propagated by chance, and that's certainly possible. We've attempted looking at epidemiologic data, which is really the only way to prove that it wasn't just a founder effect. We've attempted to do that in West Africa, in the Congo, and probably the most extensive effort with SARS coronavirus 2 has been in the UK, where enormous amounts of sequencing have been done. There have been probably several thousand introductions of virus into the UK. And so people look at transmission chains and try to calculate the efficiency. There's one big study in the UK that indicates there is some kind of advantage for the D614G, even in that context. But that all of these studies are confounded by so many variables, like the fact that the D614G came to the UK from initially people coming back from skiing in the Alps, who are going to be younger people, and the behaviors of the cohorts that you're studying are different. And what I think is clear, putting everything together, is that this virus is more transmissible from all of the experimental data, including transmission studies in animals now that are on bioRxiv. It's more infectious. It probably does not cause worse disease. It may elicit a better immune response, if anything, because of its greater openness. Maybe, in fact, be easier ultimately for a person who is infected to survive. And the good news is that the therapies, the vaccines, and the monoclonals that have been developed will probably all work against that variant. Mm-hmm. But the bottom line is, proving absolute doubt, this was not a founder effect, is really the holy grail and very tough.
Yeah.
But I don't see how you could interpret the data we have any other way.
Yep. Jeremy, this has been a great presentation. I've learned a great deal. There are a ton more questions, but we unfortunately have run out of time. Thanks very much. You were a perfect person to bring in for the Tim Gill lectureship.
Oh, thank you so much, David. And anyone, if you have questions, just send me an email. I'm happy to discuss and argue and learn from you.
Okay. Thanks again. Bye-bye.
Bye-bye.